class, we'll talk about reversible inhibitors. These are inhibitors that bind to enzymes non-covalently. So all of those great intermolecular forces like ion pairs, hydrogen bonding, non-covalent sort of hydrophobic interactions, those kinds of things. The inhibitors interfere with the activity of the enzyme in some way. We're interested in inhibitors because regulators of metabolism are inhibitors. So when we get to the metabolism section later in the course, we will talk about how pathways are regulated and how metabolites can serve as inhibitors to either stimulate or inhibit whole pathways. A lot of drugs are inhibitors. Aspirin is a cyclooxygenase inhibitor. So lots of drugs are inhibitors. So that's why we're interested in inhibitors. So we will talk about three types: competitive, uncompetitive, and non-competitive. I also have a few things to say about irreversible inhibitors, and then we'll talk about how we can control enzyme activity in metabolic pathways. So first, competitive inhibition. So if we're using the lock and key hypothesis here, where the enzyme is up here on the top in yellow, the substrate is shown here in green, and the substrate is shown fitting into the active site of the enzyme. If we have a competitive inhibitor, an inhibitor that structurally resembles the substrate so that it is actually also able to fit into the active site, then the competitive inhibitor can bind. But the competitive inhibitor doesn't get transformed to product the way that the substrate would. So we call this competitive because the substrate and the inhibitor compete for binding to the enzyme active site. So remember, all of this is reversible. So if the inhibitor dissociates, then that gives the opportunity for the substrate to bind. So in classical competitive inhibition, if we have an enzyme and if the enzyme binds to the substrate, then the enzyme can catalyze the reaction and convert substrate to product. But if the inhibitor gets there first, then the inhibitor binds to the active site, and then that prevents the substrate from binding. So the substrate and inhibitor compete for binding on the enzyme's active site, and the inhibitor structure has to be somewhat similar to the substrate, otherwise it wouldn't be able to bind to the active site, but it differs in that it can't react the way that the substrate does. So we can write our equation the way that we did the other day, where we have the enzyme and the substrate complex the enzyme and the substrate get together to form the enzyme substrate complex. Then the substrate is converted to product and the enzyme is regenerated. In the presence of the inhibitor, we form an enzyme inhibitor complex, and that precludes the substrate from binding. We have a constant called Ki. Remember the other day we lumped together K1, K to the minus 1, and K2 and called that the Michaelis constant. And under certain simpler conditions, we can consider Km to be a measure of the affinity of the enzyme for the substrate. But we also have the definition of Km in that it's the substrate concentration in which you have half maximal velocity. We have this other constant called Ki. So Ki describes the affinity of the inhibitor for the enzyme. So the lower the Ki, the greater the affinity of the inhibitor for the enzyme. So if we look at our Michaelis equation and then transform it into the Lineweaver-Burk equation, which is the equation of the straight line, what we find is that in the presence of a competitive inhibitor, we have a change in our equation, and that change is measured by this factor alpha. So you can see that alpha right here is associated with Km. So what that means in terms of our plot is that since the slope is Km over Vmax, that means our slope is going to change. It also means our x-intercept is going to change, but our y-intercept is not going to change. And what that means is that the Vmax does not change in the presence of a competitive inhibitor. So Km increases by this factor alpha. So we call Km apparent. That's the notation that we use when we're solving for the Km in the presence of an inhibitor. So what that means that when Km goes up in the presence of an inhibitor, that in the presence of an inhibitor, it takes a higher concentration of substrate to reach 1/2 Vmax. But Vmax is unchanged. We can get to the maximal velocity. If we are at a very, very high substrate concentration, then when the substrate and the inhibitor are battling it out for the enzyme's active site, the substrate can win. So this is what our Lineweaver-Burk plot looks like, and there's an animation on the Wiley website that we can look at. So we need Flash Player for that. Okay, so this is our plot in the absence of inhibitor. So in the absence of inhibitor, alpha is equal to 1. Our y-intercept is 1 over the Vmax, so we just take the reciprocal to solve for the Vmax. Our x-intercept is minus 1 over Km. So again, if alpha is 1, that's minus 1 over Km in the absence of inhibitor. If we add an inhibitor and alpha is greater than 1, then we see that our y-intercept is the same, so Vmax hasn't changed, but our Km now is, because it's in the denominator, so the Km increases, so we have a smaller negative x-intercept, so that means that we have a larger Km. When we solve for this value alpha, so our slope increases by a factor alpha, the x-intercept decreases by this factor alpha. We solve for this factor alpha because we want to use alpha to solve for Ki. So if we know the value of alpha and we know the concentration of the inhibitor, we can solve for Ki. If we have a greater concentration of inhibitor, we just have a more exaggerated look to our graph. The inhibitor concentration changes, so the alpha changes, but the Ki is the same because Ki is constant. Our Michaelis plot looks like this. If the green line is the graph without inhibitor, there's our Vmax. So if you drop a line over and drop a line down, we can, at the halfway point, we get the Km without inhibitor. If this is our plot with inhibitors, we can see that we do get to Vmax, but it takes us a higher concentration to get to 1/2 Vmax. So again, we draw a line over and drop a line down, and we can see that our Km with the inhibitor, or the Km apparent, goes up. So in the presence of a competitive inhibitor, the Vmax stays the same, the Km goes up, and the inhibitor is binding to the free enzyme. Okay, so that's competitive inhibition. Uncompetitive inhibition. An uncompetitive inhibitor binds to the enzyme substrate complex. So that means that the substrate binds first, and if it can somehow escape the inhibitor and the substrate convert it to product, then that's great. But if the inhibitor binds fairly quickly after that, then the uncompetitive inhibitor will, it doesn't bind to the active site, it binds somewhere else, but it deforms it causes a three-dimensional conformational change that deforms the active site of the enzyme. So even though a substrate's bound, the substrate can't be converted to product. So again, this is reversible. So if the uncompetitive inhibitor dissociates, then the active site goes back to normal and substrate can be converted to product. So all of this binding substrates inhibitors is all non-covalent and reversible. So here again is another picture. So here the substrate binds first and then the inhibitor binds second. The deformation of the active site isn't shown here, but the binding of the inhibitor causes a three-dimensional conformational change that prevents substrate from being converted into product. So substrate can't be converted to product even though it's bound to the active site because the inhibitor distorts the active site. So we can write our equation like this. The enzyme binds to the substrate to form the enzyme-substrate complex, and maybe the substrate is converted to product. Or maybe the inhibitor binds first, and we get an enzyme-substrate inhibitor complex. So because the inhibitor is binding to the enzyme-substrate complex, we have a different K, and we're going to call this Ki primed. So whereas K described the inhibitor, the affinity of the inhibitor for the enzyme, the Ki prime describes the affinity of the inhibitor for the enzyme substrate complex. So again, the lower the Ki, the greater the affinity of the inhibitor for the enzyme substrate complex. So our Lineweaver-Burk plot looks like this. It turns out that we have this term alpha primed, and it will change both our x-intercept and our y-intercept to the same extent. So that means over here, for this is our slope, Km over V max, we have an alpha prime term in both the numerator and the denominator, so that means they cancel out and our slope doesn't change. So in uncompetitive inhibition, the x-intercept changes, the y-intercept changes, and the slope does not change. So what happens is the Vmax and the Km both decrease, and they both decrease to the same extent, to the extent of alpha primed. So the Vmax Km ratio is unchanged. So that's why our slope is the same. And with an uncompetitive inhibitor, you can't overcome inhibition by increasing the substrate concentration because the inhibitor can bind even when substrate is bound. In fact, for uncompetitive, it has to be that way. The inhibitor has to bind to the enzyme substrate complex. So increasing the substrate concentration isn't going to help get you to Vmax. So Vmax cannot be achieved in the presence of an uncompetitive inhibitor. So if we looked at our animation here, this is our graph. So here alpha primed is equal to 1 in the absence of inhibitor. So there's our Vmax, and there's so we can our y-intercept. We can solve for Vmax using the y-intercept. We can solve for Km using the x-intercept. And if we add inhibitor, then we can solve for Ki prime. So if we add some inhibitor and we solve for alpha primed, then we can use the alpha prime value and the concentration of the inhibitor to solve for Ki primed. And again, you'll just get a greater difference between not having an inhibitor and having an inhibitor if you have a greater amount of inhibitor. That will change the inhibitor concentration and the alpha primed, but not Ki primed, because Ki primed is a constant. So this is what the Lineweaver-Burk plot can look like. So if in black, this shows a normal graph, and we look at the halfway point and draw a line over and drop a line down. If the curve looks like this, then there's our Vmax, and we will have a different halfway point. So we drop a line over and drop a line down, and then we can see that our Km decreases. So the Km decreases and the Vmax decreases to the same extent, and that extent is alpha primed. Okay, third type, non-competitive inhibition. It's sometimes called mixed inhibition. Here, the inhibitor binds either to the enzyme substrate complex, or it can bind to just free enzyme. So either inhibitor or enzyme can bind first. And it's the same kind of strategy where when the inhibitor binds, it deforms the active site so that even when substrate is bound, the substrate can't be converted to product. So whereas a competitive inhibitor only binds to enzyme, and an uncompetitive inhibitor only binds to enzyme substrate complex, a non-competitive inhibitor can bind to both free enzyme and the enzyme substrate complex. So here, if we look at our graphs again, the inhibitor can bind to free enzyme, or the inhibitor can bind to the enzyme substrate complex. So an enzyme substrate inhibitor complex is possible. But what this means is now we are going to need two different Ks. We need the Ki primed that describes the affinity of the inhibitor for the enzyme substrate complex, and we need Ki, which describes the affinity of the inhibitor for free enzyme. So for this type of inhibition, we need a Ki that describes the affinity of the inhibitor for just the free enzyme, and a Ki prime that describes the affinity of the inhibitor for the enzyme substrate complex. So this is what our equation looks like. We have both alpha and alpha primed terms. So the slope changes by a factor of alpha, the y-intercept by a factor of alpha primed, and x-intercept is affected by both. So in this type of inhibition, Vmax always decreases. And again, this is a situation where even at high substrate concentration, the Vmax can't be achieved. Well, what about Km? You know, if we look at the x-intercept over here, the Km, depending on the relative values of alpha primed or alpha, Km might increase, it might decrease, or it might stay the same. So let's look at that. Here's an example of a non-competitive inhibition, and in this case, alpha is greater than alpha primed, which means the Km goes up. And when that happens, the inhibitor prefers free enzyme. So we have another animation here. So again, here's our plot of our enzyme catalyzed reaction where alpha equals alpha primed and they both are equal to 1 and there's no inhibitor. We solve for the y-intercept by looking, we solve for the Vmax by looking at the y-intercept. We use the x-intercept to solve for Km. Now if we add some inhibitor, and here this is an example where alpha is greater than alpha primed. In that case, this is the appearance of the graph. The lines will cross above the x-axis. You'll have a smaller negative number for the x-intercept. So because Km is in the denominator, that means that Km increases. And our y-intercept is higher, indicating that the Vmax has decreased. And again, just in the other animations, we'll just get a more dramatic difference between without inhibitor and with inhibitor if we add more inhibitor. We can solve for Ki by using the inhibitor concentration and alpha. We solve for the Ki primed by solving for alpha primed first, and then we use the inhibitor concentration. So our graph might look like this, where if we look at our plot without inhibitor, and then we do our 1/2 Vmax, which would be here, drop a line over, drop a line down, there's our Km without inhibitor. Whereas if our plot with inhibitor looks like this, there's its Vmax, so half of that brings us up here, and now we have the Km with inhibitor. So that's the Km apparent, and it is greater. So this is the situation where alpha is greater than alpha primed. In this situation, Km goes up when there's inhibitor present. That means that the inhibitor prefers enzyme. The inhibitor has a greater affinity for free enzyme than it has to the enzyme substrate complex. And the appearance of this graph will be that your lines will cross above the x-axis. Now suppose we have a situation where alpha is less than alpha prime. So if alpha is less than alpha primed, then what happens is, in this case, the Km decreases. And the appearance of the plot is that they will intersect below the x-axis. So in this case, that means that the inhibitor prefers the enzyme substrate complex over just the free enzyme. There's a guided exploration that we can look at this, and you'll have to enable the Flash player. So this is kind of fun to look at, to use these little sliders right here, and you can vary the Km, you can vary the Vmax, you can add a competitive inhibitor, you can add an uncompetitive inhibitor, or you can add a mixed inhibitor. So now here the situation is Ki is 2 times Ki prime, so they're only giving us one of those examples. But if you want to play around with this to understand how these things are affected, that might be a good exercise. Okay, so here is our last possibility. So in non-competitive inhibition, the inhibitor can bind to either the enzyme or the enzyme substrate complex. So sometimes the inhibitor prefers the enzyme. Sometimes the inhibitor prefers the enzyme substrate complex. Sometimes it doesn't have a preference. The inhibitor binds to the enzyme and the enzyme substrate complex with equal affinity. So when that happens, that's the appearance of your graph. What will happen is that they will, the lines will meet, in the at the x-axis. And then this is what the plot looks like. So when you do the 1/2 Emax here and the 1/2 Emax here, then you see that the Km doesn't change. So here is a summary of all of these equations. Here's the Michaelis equation, here's the Lineweaver-Burk equation. This shows the effect of the inhibitor. In this, the authors here use this Km apparent or Vmax apparent. Sometimes I've seen Vmax with an I. It means the same thing. That's the notation that you use that when the Km or the Vmax changes in the presence of the inhibitor. In our manual, on the last page of the very last page of the manual, on one of the green pages, I've taken the Lineweaver-Burk equations and I have presented the slopes, the x-intercepts, and the y-intercepts, which make it easier for you when you're trying to solve these kinds of kinetics problems. So as an example, I thought I would give you succinate, which is a substrate, and malonate, which is an inhibitor. So hopefully that you can see that either one of these can bind to the enzyme. Oops, sorry about that. But when the succinate binds, then the succinate is converted to fumarate. If the malonate binds, then you get no reaction. So these are the characteristics of a competitive inhibitor because, and we know that this is a competitive inhibitor because the structure of the malonate looks a lot like succinate. There's just a difference of one methylene group. So competitive inhibitors structurally resemble their substrates. Okay, so I just wanted to say a little bit about irreversible inhibition. So everything up until this point has been reversible. So our competitive, uncompetitive, and non-competitive inhibitors are they all bind to their enzymes non-covalently, which means they're reversible. They can bind and then they can dissociate. If we have irreversible inhibition, that means there's a chemical reaction that takes place and there's a covalent bond forms between the enzyme and inhibitor. And if you can't reverse that reaction, then you usually destroy your enzyme. So this is an example with chymotrypsin, which has a serine. We'll find out when we get to chapter 6 that this serine right here is essential for activity. You have to have that OH group, and if you don't have the OH group, the enzyme is inactivated. So this compound right here, diisopropyl fluorophosphate, we can carry out a chemical reaction with this, and then you have the enzyme with this large group attached to it. You no longer have the OH, and since the OH is critical for enzyme activity, the enzyme is inactivated. This has been used as an insecticide. If you treat crops with something like this, you can interfere with nerve transmission of an enzyme called acetylcholinesterase, which is has a mechanism very similar to chymotrypsin. But it can also be used in chemical warfare. So when you hear of various nerve agents, this is one of them. So the reaction with the OH group and the acetylcholinesterase interferes with nerve transmission. So let's go on and talk about how inhibitors can be used to regulate enzyme activity. So here's an example of an inhibitor, phosphoenolpyruvate. So it's a metabolite in one of the later steps in the 10-step pathway glycolysis, which we'll talk about later on in the semester. This metabolite can serve as an inhibitor of an enzyme that catalyzes an early step in the process. And in this way, the phosphoenolpyruvate can regulate the pathway. So there are two forms of phosphofructokinase. Maybe it would be nicer not to have them superimposed, but this is all I have, unfortunately. We're going to borrow some nomenclature from hemoglobin, where we have T and R forms. So the T form of the enzyme is shown here in red, and the T form has a low affinity for substrate, just like the T form of hemoglobin has a low affinity for oxygen. The R form of an enzyme has a high affinity for substrate, just like hemoglobin in the R form has a high affinity for oxygen. So if we can just focus our attention here on the green, this is the R form that's shown in green. This is the active site. There's a positively charged arginine, and there's our substrate, which is fructose 6-phosphate, which is negatively charged. So this very favorable ion pair forms between the substrate and the enzyme. Well, what happens over here, also shown in red, when the phosphoenolpyruvate inhibitor binds, there's a conformational change that occurs. And what happens then is this arginine is now moved over here more into the interior of the protein, and now this glutamate swings over here into the active site. So now, instead of having this favorable ion pair between the enzyme and the substrate in the R form, in the T form, the conformation of the enzyme now has a negative charge in its active site, which repels the substrate. So this explains how phosphoenolpyruvate is able to serve as an inhibitor. And we can see this if we go into the lab and we can look at this graph where we can see that the Km of the enzyme without the phosphoenolpyruvate inhibitor is much lower than the Km of the enzyme when the phosphoenolpyruvate inhibitor is there. So again, when the Km increases, Km is defined as that substrate concentration at which the velocity is 1/2 of the maximal velocity. So in the presence of the inhibitor, it's going to take far more substrate to reach half maximal velocity than if the inhibitor were not present. So the other thing I hope that you noticed is that this curve right here is sigmoidal. So, and that is because allosteric enzymes are usually multi-subunit. So remember, hemoglobin is an allosteric protein. It's multi-subunit. So allosteric enzymes are also usually multi-subunit. And there's cooperativity involved with these types of enzymes binding to their substrates. Just like for hemoglobin, there was cooperativity involved in the oxygen binding to the hemoglobin. But unlike hemoglobin, we have two models. We have a concerted or a symmetry model and a sequential model. So different enzymes use different types of models. So if we can use just sort of this cartoony drawings right here, if this is a dimer and we have two subunits, so each subunit has an active site, and imagine that you have a substrate that fits very nicely into this active site. So this would represent the R form of an enzyme with a high affinity for the substrate. And again, if you're imagining a substrate that can fit into there very nicely, if this is the T form, then the substrate doesn't fit in there very nicely. So the T form has a low affinity for substrate. So we're using the same kind of nomenclature here for enzymes that we do for hemoglobin. So in the concerted model, what happens is that if I have my dimer that's in the T form, it's difficult for the first molecule of substrate to bind because that molecule of substrate binding needs to drive the conversion from the T form to the R form. In the concerted model, the subunits of the protein all change together in concert from the T to the R form. That makes it much easier then for a second molecule of substrate to bind. So in the concerted model, you have either all the subunits are T or all the subunits are R, but you don't have any kind of intermediate configuration. That's shown here in another figure from another textbook, where the substrate can bind to the subunit in either conformation, and that the T and the R states are in equilibrium. But when the transformation from the T form to the R form occurs, it happens in concert all at once. Some enzymes that are allosteric follow the sequential model. So here again, this substrate has to drive the conversion of one of the subunits from the T to the R form. This subunit stays in the T form, but because this is a dimer, there's communication that occurs between these subunits, and that allows for a for an easier conversion for this T to R, and then the second substrate of molecule of substrate binds more easily. So we still have cooperativity here as well. So here's another picture of the sequential model. So in the sequential model, the substrate binding progressively induces conformational changes. So the greatest changes occur in the subunits that already have bound substrate. So in the sequential model, we do have T and R forms of different subunits allowed in this model. So because we have these R and these T forms, this explains what we saw with the phosphofructokinase and the phosphoenolpyruvate. We can have allosteric activators or allosteric inhibitors. So in my phosphoenolpyruvate example, the phosphoenolpyruvate was an allosteric inhibitor. It bound to the enzyme, not at the active site. It bound somewhere else, but it caused a conformational change that altered the structure of the active site so that the substrate can no longer bind. This is similar to what happens in hemoglobin. We saw that carbon dioxide and protons in PBG all stabilize the T form. But we also have allosteric activators, and again, they bind not to the active site, somewhere else. That's what allosteric means. It means other place. And when they bind, they stabilize the R form. So allosteric activators stabilize the R form, and allosteric inhibitors stabilize the T form. So I wanted to close with this example. This is a pathway that leads to the synthesis of CTP. CTP is a pyrimidine. One of the many ways that the cell has of ensuring that when DNA replicates itself that few mistakes are made is that it maintains equal pools of CTP, GTP, ATP, and TTP. There are other mechanisms too, but this is one of them. And the pathway starts with carbamyl phosphate and aspartate, and ATCase is the enzyme that forms N-carbamyl aspartate. And then there are a number of steps that you get until you get to the product. But the way that this works is that if the CTP concentration gets to be too high, then you'll have the 4 nucleoside triphosphates will be unbalanced. So the CTP is a product, but it also can act as an inhibitor for this first reaction. So here's a picture of how this works. So up on top, we have, this is the enzyme. It's a hexamer. So we have these catalytic subunits shown in blue and green, and they have to have a certain conformation in order for the substrate to be able to bind. But the enzyme also has these regulatory subunits that are shown in yellow, and the CTP here, there are 4 regulatory subunits, and there's a molecule of CTP bound to each one. So CTP is stabilizing the T form. Another way to look at that is like maybe this is kind of like a stool with the kind of crooked legs. So this will be the situation if the cell has ample amounts of CTP. The ATCase will remain in the T form. But say the CTP levels drop. Well, when that happens, the CTP will dissociate, and when the CTP dissociates, the legs of our stool straighten out, and now these subunits come together in a way that forms the active site, and now the enzyme is on, and the enzyme is in the R form. So this is not shown in this figure, but ATP actually favors the R form. So there are binding sites somewhere on this regulatory subunit that stabilize the R form. ATP being a purine. So if ATP levels become high, ATP is going to want to turn on this enzyme to make sure that the CTP levels come back up to the levels of the ATP. And we can see that in this figure right here. So I think the colors were not chosen well. So in this bluish purple right here, this is the curve that we get when we measure the velocity and we have increasing concentrations of one of the substrates, the aspartate. So there are no allosteric effectors. And again, this is a multi-subunit enzyme, so we get a sigmoidal shaped curve. That means those Michaelis-Menten equations don't work because those are equations of a hyperbola, but we can still use the concept of Km and Vmax to figure out what the Km is. So I would not have used green, I would have used red here because CTP is an inhibitor. So the Vmaxes are the same. This is just with an experimental error. They don't show the error bar, so it's not clear, but the Vmax up here is meant to be the same at high substrate concentration. So if we look at the Vmax and then look at the Km, we can see that the Km increases in the presence of CTP. CTP is an inhibitor. It stabilizes the T form. Km goes up because in the presence of CTP, it takes a greater concentration of substrate to reach half maximal velocity. So we can look at this. So for example, if we had 8 millimolar aspartate, and we looked at 8 millimolar here in this blue line with no allosteric effectors, and if we looked over here where CTP is present, that flashing red arrow indicates the amount of the decrease of the activity that you get in the presence of CTP. So again, I wouldn't have chosen red. For this one, I would have chosen green because ATP is an activator. So ATP is a purine, and ATP, if it becomes high, that means purine concentration is high. So we want to increase the concentration of the pyrimidine, so we should activate that ATCase enzyme. And again, if you look at the Km, you can see that the Km goes down. So that means that less substrate is required to reach half maximal activity. And here, if we use the same kind of analogy, again, if we look at 8 millimolar aspartate, and if we look at this flashing red arrow right here, this arrow indicates the increase in the activity that you get when ATP is present. So the other thing that I hope that you noticed is that the ATCase is an example of the concerted model, because when you go from T to R, the subunits all change in concert, or they change together. So the ATCase is an example of the concerted model. So that is our lecture for today. Here are my references. I hope this was useful, and I will see you next time.